It's a great pleasure to open the summer school in Theoretical and Computational Biophysics. I know that we have an easy time with you because most of you just came for the beauty of the surroundings of Urbana, so you probably don't expect too much here. Not joking now, the school is actually brought about through an important initiative of the National Science Foundation. And that's an initiative that goes a little deeper than what you call usual program announcements. It's an all-pervasive aspect of funding in American science nowadays, and that is that scientists who are pretty well treated since the last 50 years, if you compare that with the Middle Ages or some other times, then we cannot really complain. But the taxpayers and the people who channel from the taxpayers the funds, the scientists feel that it is actually a great privilege to be a scientist, and we scientists work hard for the funds we get, but the taxpayers in America and the people who organize our country feel that scientists should give back to society. And the National Science Foundation called it outreach, and we are all asked to do our share, and we strongly believe here in our group and at the Beckman Institute that we should do so, and because of the generosity that is bestowed on us by the American taxpayers, we feel strongly that we have to give something back, and so this workshop is actually an opportunity for us to do that. We are a well-funded enterprise, we have lots of excitement with our work, but we feel that we should share that, and so you will hopefully agree that we have tried hard to do that. Now, before I also continue, I have to explain the word we, and that is actually a group of young people, of not so young people, of students and postdocs and faculty, but particularly also of staff who have spent an incredible amount of time to organize this workshop. It's one of these situations where you really don't know what you're getting into when you say, okay, we do it in 3 years from now. That's what we said 3 years ago, and then it exploded on our heads. And so there are people, I just show here the group, some of the students, not the faces necessarily, but you see that there are all those who have done something. There are some other students who have worked very, very hard, and literally we haven't, many of us are sleep deprived since a few days, getting everything finished. And so I really have to point out to you that it is this group of people who really believe in doing something for the benefit of others, because we are privileged of being at this beautiful place here and doing our science, and so it's us who is trying to teach you now the next 2 weeks. But I also have to say something warm and nice about you, because we really feel particularly excited about what we are doing the next 2 weeks, because of the great resonance that we got when we announced this workshop. We were actually scared that nobody shows up and nobody is interested in it, and the opposite was true. It was a little bit difficult for us to be in a situation that we had to say actually no to quite a few people, and so we are very glad that there is interest, and of course we understand that it is not so trivial for you to come, some of you from pretty far away, from abroad, from Asia and from Europe, but also from many parts of the United States. And we feel very honored and thankful that you feel that you trust us and that you come here to learn from us. I'm pretty sure, having seen already some faces from well-known laboratories, that we are also going to learn something from you. And that's usually the nicest situation, that one learns from each other. So let me then quickly give you an overview of the workshop. I would like to start by introducing the place. First, the University of Illinois. We are very proud of our university. It's actually a state university that is one of the few state universities that is really in the top rank of American universities research-wise. You will see that despite of maybe some bad name we have, that's actually a very beautiful town and a very beautiful campus. You have seen when you marched up here to the Beckman Institute that everything is shiny and new, because it's a very dynamic place. And you're here on the engineering side of the campus that has been built only during the last 10 years. And so I hope you will find your way around the university, and I'm pretty sure you will enjoy it. The weather is usually nice around this time of the year, and so you might actually also may want to wander a little bit further afield, and you will see that we are land-grant university, and one of the nice things is actually that we are agricultural. That is not necessarily disadvantage. I personally think it's just very beautiful to walk around. I also would like to say something about the Beckman Institute. The Beckman Institute is one of the first American interdisciplinary research institutes. It's now 14 years old since we moved in here, and it is actually a United Nations of science and engineering, meaning that the groups who are here do not belong necessarily to one department, like the Theoretical and Computational Biophysics group has students from physics, from chemistry, from the life sciences, from computer science, sometimes also from engineering, and everybody feels equally welcome and happy, and no particular field dominates a culture. And that's a very good approach to many of the problems that science and engineering faces today. It's also a place where there is no constructed difference between pure science and applied science. It's an institute for science and engineering, and so we really benefit from combining both. And you will probably see during the 2 weeks that the technical expertise that we can bring to the fore in doing science, and for example giving you opportunity to do your own numerical experiments in the laboratory that we provide you with, that is actually an expertise that is very rare and can only be found in places where engineers and scientists mesh very well. I'm proud of the university also because of our great tennis team. I don't know how many of you play tennis, but they won all 3 national titles this year. And so it's a great pride for us to have such wonderful young people here. Now I would like to run you briefly through the program. We have a program that could be misleading, and I have to explain to you immediately something about the program and the character of the workshop. And that is that the most important part of the program is actually the afternoon and the evening, when we have hands-on sessions. We have written almost 300 pages of laboratory tutorials for you, which is why you might find some of us a little bit boring and tired, like myself. I had very few nights of sleep, and the last night not at all, because we wanted to finish these tutorials, write them in a way that they are very clear and not too error-prone, and even if you make a few errors, that you might still be able to continue, because we really feel that the way to learn computational biology is through doing it rather than through talking about it. And so you might find that we are a little bit, I don't want to say weak, but we do not emphasize the theoretical peaks in our morning lectures, because we want to provide the lectures in a format that really bridges to the afternoon and evening sessions. And since we have to count on quite a few of you being novices in the field, we are not aiming at having very complicated examples, and so by necessity everything is a little bit down to us. Our lectures will be, even though several of us are mathematicians and theoretical physicists and work in quantum field theory and in many electron physics and so on, but we really try to keep ourselves back and try to be as simple as possible rather than as elegant as possible, so that we really are serving the purpose of a hands-on approach to the field. So the work that we have done for you is really in the tutorials, and you will find, actually that's not on the slides that I show you, that in each of the lectures as we go on, you will find that the lecture material, usually however only in terms of PDF files that we take from our PowerPoint presentations, will be available. But in the afternoon sessions, you will find for all of them the tutorial material. Like for example, today we have a VMD tutorial. VMD is a graphics program that we will be using for molecular graphics purposes. That's about a 50-page document that we wrote for you. Tomorrow and on Wednesday you will find two tutorials, one on the program NAMD for molecular modeling and another one on calculating potentials of mean force with steered molecular dynamics, and so on and so on. So for the whole week we have these tutorials written, but even the writing, that was hard. It's about, as I said, 300 pages. That wasn't even the difficult part. The difficult part was to make sure that it works in the laboratory. And we tried hard. I'm pretty sure there will be many problems, but I can tell you that during the last few weeks there were many catastrophes and surprises and little glitches here and events there. And we hope we went through many of them so that you can actually enjoy doing actually the work rather than running constantly into problems. But as you know, there's Murphy's Law, and we are only human. There is very likely, it's very likely that there are problems left. And so I think it will be very good if we work together here and see that we are circumnavigating them by you calling attention to the problems quickly and so on. Now in this regard, I have to make another important comment, and that is really a comment that is very crucial to the success of this conference. More so probably than all the lectures we give and all the preparations we did. And this is something we have to count on people helping and doing something, and that is you. And I mean this. The distribution of participants we have, we wanted to be sure that there are also quite a few among you who really do not know the field yet, is very broad. And it is very likely that some of you have initially at least trivial problems. And so I really ask you, I hope you will realize very quickly that we worked very hard making this possible, and we really worked for the purpose of this conference, not just pulling out of drawers lectures that we had ready. And it would be very nice if you, in kind, not to us, but to your colleague, give this back by really distributing yourself, for example, in the laboratory, so that the experienced one are meshing a little bit with the inexperienced one, so that people can ask the neighbors, and it would be very nice if we have a culture and spirit of collegiality and of being helpful, because I think that way we can really be much more successful. And as particularly the more experienced among you will probably know, you learn a lot from explaining something to others. And so you might want to take the opportunity to learn more and learn things in a more robust way by explaining it to the young man or young lady next to you. And so please, let's try to keep that in mind. I think this kind of hands-on work is not possible without the kind of tutoring that you find often more from student to student, or in this case participant to participant, than from teacher or faculty to student. So last but not least, let's now go through the program. So the first day we will be talking about the simple aspects of modeling, the graphics and the basics of molecular dynamics. And then in the afternoon we have the first hands-on session, and we have to be initially a little bit out of sync with the lectures, because it is not possible that you are learning so quickly from zero that we can have you be in sync with the lecture. So this afternoon we count on it being more expedient that you just learn molecular graphics. Now, those of you who know something, don't wave your hands, because we really want to not only show you basic molecular graphics, how to rotate a protein around to load it, but also the scripting behind it and the more elegant methodologies connected with scripting. And so I'm very sure that also the experienced among you will get some benefit out of it. Now in the evening today, not tomorrow because we have a little party together, but then all the other evenings, the evening you have basically time where you can do your own work. You may just try to learn more if you didn't get through all the material in the afternoon. But you can also try out your own new examples for the material that we propose, but it's also possible that you just do your own project, that you let yourself be inspired doing some work that you might have done anyway at home. We brought to your attention that you can bring your pet protein along, and you might then in the afternoon, in the evening, work on your own project. We of course cannot give you huge computer time. We got a generous amount of computer time, but it's not like huge that you can do some incredible calculation here. But you can definitely do something respectable. And those of you who want to take advantage of this, they are invited then to what we call the beauty contest. We ask you then to enter your entries to us Wednesday morning. We decide then in the morning which of the entries we will have then for lectures Thursday evening. And then Thursday evening we will then have, I don't know how many, it depends a little bit on the entries, but we'll have a few lectures by participants on your pet project, on the beauty of your own biomolecular system. And then we will have a little contest through applause. We measure the level of applause and see who you think did best, and we hope we can come up with a little prize for that person. So this is the evening, and you are pretty much under pressure time-wise, because we have only limited time in the laboratory. That has something to do with the fact that we needed very good workstations, and we had to go to one of the computer laboratories of the College of Engineering here at the university, and they are staff and they don't want to be around all the time, so we had to actually pay for it a little bit, not much, but we had to. And so you have 2.5 hours in the afternoon, 2 times 1 hour, 50 minutes, and then you have 2 hours in the evening. It might be stretchable if some people volunteer to look after the laboratory, but at this moment we cannot promise it yet. So we will see a little bit how it develops, and we hope very much that the time is sufficient. You have then, 4.5 hours of hands-on work, and when we went through the material ourselves, we were pretty tired already after a single hour. So maybe 4.5 hours is actually enough, and you might want to do something else with your time anyway. But anyway, that's a little bit of a limit. You should keep it in mind so that you get through the material. You should again remember that it is actually the hands-on that probably is the greatest value for you. And probably most likely, most of you will say this was really a good thing of this workshop, and glad that I did my exercises rather than go to the bars. So that's the first day. All the other days are pretty similarly structured. On the second day, we talk a little bit more about more advanced general behavior of biomolecular systems, so-called non-equilibrium behavior, and properties that can be simulated and analyzed. And then we have in the afternoon, Tuesday afternoon, the tutorial for molecular modeling in connection with an MD program. And so there you get then a fat tutorial on MD, 100-page tutorial. And that tutorial you also work on the next day, on Wednesday afternoon and Wednesday evening. And on Wednesday morning, we talk about the methodology that is called steered molecular dynamics, where one actually manipulates processes in proteins to accelerate them, to try them out. And this is sort of like real fun to play around with proteins, and we hope very much that you will enjoy this, hearing about it and trying it out yourself in the laboratory. And on Thursday, we talk about a very timely subject of modeling on membrane channels. Since we are here in a school where we learn the basics, we will talk about very simple channels, namely water channels. And in the afternoon, you have then a tutorial on membrane water channels. On Friday, we have then the first session on quantum chemistry. Almost invariably, when you are studying a process in a protein, an enzyme, or any other biological system, and you are focusing and want to understand things in detail, you come to the realization that the simple force fields that are being used are not good enough, and so you are then in the situation that you need to improve certain aspects of it, and for that you need quantum chemistry, and we have 2 days devoted to this. On Friday, you will have the first lecture on quantum chemistry with the hands-on in the afternoon, and then on Monday the second one. But before the Monday comes is a weekend, and on the weekend we have not anything planned at this point. Maybe we will plan something, but this is not so clear yet. But on Saturday, we definitely want to give you an opportunity to see the surroundings, and the most interesting surrounding we can think of here is Chicago. So we have rented a bus that goes with you to Chicago. However, you have to pay for it, so we rented it for you. We have only limited number of seats, and so you are advised to sign up for it. We have to go first come, first serve. Some people have already signed up through email, and so now I think there are about 20 places left. And so if you want to go to Chicago on Saturday, you have to sign up. I understand it costs about $30, and the bus will leave in the morning, I think at 8, and it will return about 10 o'clock in the evening, and so you have a nice day there. And Chicago, if those who know it can tell you, it's a beautiful city. It has a waterfront and shopping and great food and great museums, and so you might have a good day there. So on Monday we do then the second part of quantum chemistry, where we focus on how to improve force fields, the parameters in force fields, and we have a hand-on then where this methodology is used for a certain enzymatic system. And on Tuesday, you learn about bioinformatics methodologies, sequence analysis, multiple sequence analysis. That's a methodology that is an absolute necessity today for a good young researcher and also an old one, because of the great information we have through the sequencing efforts. And so you learn how to weave this into your modeling. On Wednesday, we talk then about two aspects. One is on parallel computing and one on modeling of membranes. And we have there our first guest speaker. Actually, we have one guest speaker, but it's almost family, Johan Kostin, who talks about non-equilibrium processes on Tuesday. But then on next Wednesday, we have Mike Klein from University of Pennsylvania, who talks about a new method of doing lipid simulations. And in the afternoon of Wednesday and Thursday, you will have the opportunity to learn parallel computing by hand. Namely, we are going to give you the opportunity to build a cluster. There will be a lecture Wednesday afternoon introducing cluster computing, and then we have one session of actually building really a cluster of workstations that are then all being networked. You do this one together with some of our co-workers here, and there will be 3 sessions like this, one on Wednesday and 2 on Thursday afternoon. So on those days there will be no hands-on session, but we rather build a cluster. Those of you who come into the situation of suddenly being fortunate of getting a faculty position, you are post-student, you are postdoc, and then you're getting an offer for assistant professor position, then you know that you have suddenly a big problem on your hand. You have to suddenly build your own computer lab. And one of the things you have to do today is build a cluster, because that is how you can do community computing today. And you can learn here how this is done in one example, and brushing once with this kind of task will make it much easier for you to do it the next time. You will realize it's actually doable, and you have to know a little bit, but it's not the end of the world to do it. So this will be our Wednesday and Thursday afternoon activity. And I mentioned already that on Thursday we have then our beauty contest. So again, in the evening you might want to work on your own projects, and you can then, you will see that we give you examples, but you may just also use your own example, then you present them on Thursday. On Thursday we also have the third guest speaker, Tamar Schlick from New York University. Tamar is the author of a recent textbook in computational biology, a wonderful book I can recommend very strongly, very personable written, engagingly written book. And she will talk about some important numerical aspects of modeling together with her local colleague here, Bob Skeel. And on Friday, we come then to one of the expertise of the Illinois modelers, namely large system modeling. Since you cannot do any much hands-on afterwards anymore, we feel that might be a good subject to enter the summer school with. So we will speak then about photosynthetic systems, where we have on one side large protein aggregates, on the other side we have some interesting quantum processes, and the afternoon session in this case is wonderful for the physicists and chemists at heart among you who want to learn a little bit about what is often called quantum biology, the quantum processes that are realized in living cells. So that is a program, and we also provide you with a tool that is called BioCore, that some of you became already familiar with because we advertised it to you already before you came here. And BioCore has several functions here. One is a social function, so it's basically a communication program that permits you to talk to other members of this workshop. It's what is called in modern language a collaboration software. It is project-oriented, and one project is a social project. We form a social group here, and you can then exchange suggestions where you want to eat and which bar you like, or where you went bicycling or walking or whatever. And so that you can do. You can also have a chat window for problems you encounter in the lab, or suggestions you have, or questions you have about your work. And so this BioCore is, once you're getting used to it, it takes a little while, but many of you are probably already over it. It doesn't take that long, but I just want to encourage you, just take a little energy to try it out a few times, and then it becomes actually a nice routine tool to communicate, and that might actually make us all a much closer learning community than without this program. So now I come to some last general comments. So I would like to stress again that the summer school is a volunteer effort. We are no professionals. We're doing it for the reason that we think that outreach is an important component of science in the United States nowadays. The main focus are the hands-on sessions. And the aim is to get you to do, that's the most important word here, computational biology. We have provided for you tutorials, and the optimal course that this all works is that you help each other. So you are playing also a very important role in the afternoon, the evening, that you are looking over each other's shoulders and try to help the next fellow. The beauty contest I mentioned already. I also mentioned the trip to Chicago. I would like to just end up by asking you for two types of feedback. One is what you may call the critical feedback. We have the sessions in the laboratory, the lectures, and the tutorials in particularly, and we want these tutorials to, of course, all the way we are doing this school, we want this to become better. And so we ask you to write revisions and suggestions into the tutorial. We will be providing the tutorial to you, but also we will put in each of the computer laboratories a copy of a tutorial, so that you can go there and write, real terrible or nice example, or here's a sign error, or there the file is mislabeled, so that we improve eventually these tutorials. And so this would be very nice if you could actually be a little critical in a constructive way, but also in a non-constructive way, whatever you feel like, so that we really, at the end of this workshop, can go then to the notes you gave and improve the tutorials. We also put the tutorials here the next morning. So let's say the Thursday tutorial will be here Friday morning, will be on the laboratory, but will be lying here so that you can put your notes into them. We also like you for what you may call the more positive feedback, in the sense that we of course have to tell the National Science Foundation how things work, and if this should be done again. And so if you feel like that, you might then at the end of this 2 weeks say, or send us back an email, say this was actually something you should be done again. It is encouraging, so that the National Science Foundation knows that this was a worthwhile effort. So now let's enjoy 2 great weeks of learning and friendship. So I'm done now with my first welcome. Probably I was even stayed reasonably in time. I forgot one other thing as I was running through the program quickly. We have on each, at the end of each morning, we have 15 minutes question-answer period, where you can ask any kind of question, but probably better, you know, science and method-oriented, but you can also ask about food or something. So that you will have a chance to have a discussion here for 15 minutes. Of course, it can also be a little longer if there's some heated point. And so please keep that in mind. So I think the lectures go to 12:30. From 12:30 to 12:45, I will try to see that I bring quite a few of our team here to be present, so that we can give you immediately some good answers. Okay, so let's breathe then. You give me a minute or two to change my PowerPoint presentation, and then we will begin with the first lecture.